Today we have 2 presentations, which is exciting, and we will have a fun little activity, like a little Kahoot, which is just like research-based, just because we wanted you guys to be able to like do something fun that we would probably normally do if we were in person. And then we'll give you some details about the presenter coming up, and yeah. All right. Oh, it's the 9th, obviously. Yes. Okay. So groups 2 and 4 are presenting today. Let's start with group 2, if that works with you guys. Let's see. How do you want to do this? Do you want to share your screen, guys?
Yeah, sure. I can share my screen.
Okay. Jasmine, do you have to give them like co-host things or whatever? Perfect. And then just guys, really quickly, whoever else is here, just while we're listening to this other group's presentation, just in general, make sure you write down any questions you have, be an active listener, and like note any forms of constructive criticism, just everything that you do normally in that sort of presentation. So go ahead whenever you guys are ready.
Okay, so can you guys hear me?
Yes.
Okay, so hi, we're group 2. It's me, Samiha, Maheen, and Chaith Raleigh actually had a class right now, but she did do her slides, so we're gonna just read them for her.
Okay, thanks. Sorry about that.
Yeah. So our research was about the implantable wireless device platforms for neuroscience research. And it was novel research conducted by Philip Guttruff and John A. Rogers. So for the introduction, this article basically explains the emergence of ultra miniaturized wireless devices that have a variety of different uses. This device was, while only tested in mice, it shows a lot of promise in delivering medicine straight to the brain, stimulating via optogenetics, and as a way to measure physiological data. It consists of next generation multifunctional platforms that can be used for a variety of different purposes. And the device shows great promise in research as well as healthcare. The significance of this device in particular, it has a lot of intellectual significance in academic research as well as practical value in healthcare. The most promising area, which is covered like in a lot of depth in the article, is in the field of optogenetics, which is the use of light to control neurons that have been genetically modified. This device can be used to test different areas of the brain for irregularities, and it's the first step to detecting neurological disorders earlier in their development. And the ability to deliver medicine directly to the brain is really like a pretty big milestone, especially when it can be left in and doesn't cause harmful effects. And this is Chaith Raleigh's slide. So basically this is her explaining the platform. So it's multimodal probes for the delivery of light, fluid, and electrical current. And an important goal of this new neuroscience research is for this miniaturized, fully implantable platforms with wireless capabilities and battery-free operation. And this helps to avoid the limitations that characterize the conventional approaches. And this device is sensitive enough to remain fully implanted in the animals of all sizes without harsh side effects. And to address this, these recent engineering developments have greatly expanded the scope of the capabilities in neural interfaces. For example, platforms based on optical fibers as simple waveguides to transport light for optical stimulation have evolved to multimodal probes for the delivery of light, fluid, and electrical currents. Other notable devices include syringe injectable electronics that unfold and target tissue to record electrical signals, ultra-thin organic transitor arrays for the situ myoelectric signal recording of optogenetically evoked spikes in muscles, and multifunctional electrodes for recordings in non-human primates. And Maheen, this is where you take over.
Yeah, can you guys hear me?
Yeah, we can.
All right, this is also Chitrali's slide, so I'm just going to read out what she had told us to. So, battery-powered devices, while effective, have some challenges, including the fact that they consume significant space. They are heavy and often involve toxic materials, and they offer limited operational lifetimes. Wireless power transfer schemes implanted with miniaturized circuitry and specialized antenna designs can circumvent limitations placed by the battery power devices. The most recent generation of wireless devices for optogenetics include optimized antenna designs in the radio frequency, which allow for efficient transfer of energy to power highly miniaturized light sources. As illustrated in Figure 2a, a typical device can be injected into the brain of a small animal model with minimal impact on the host due to the small size of the device. Figure 2b shows a similarly miniaturized device with place aversion and spatially and optical separated stimulation of the ventral and dorsal regions of the nucleus. Another different but related device shown in Figure 2c uses hand-wound coils to supply high field densities. The use of such devices is limited to custom-designed experimental areas that can be difficult to adapt for varied experimental. For example, these devices can be used to create stimulations of the motor cortex via optical elimination to yield changes to locomotion of various experiment animal groups. Lastly, devices that analyze neural circuits can also be used to administer liquid drugs. Figure 2d shows a wireless device for stimulus optical and fluid delivery with demonstration of drug-induced rotational locomotion via optical stimulation. This technology has relevance in other contexts, such as the alteration of gene expression within animal subject studies. This shows the universal nature of these devices because they can be applied and used in many different areas of research. It's not limited to neuroscience. So Mia, can you go to the next one? Thank you. Applications of optogenetics also offer promise for dissecting in neural circuits of the spinal cord and the nervous system, with additional goals for the reduction of pain, stimulation of paralyzed muscles, and treatment of cardiovascular dysfunction. For this objective, miniaturized wireless soft devices are highly beneficial because they provide a platform with minimal impact on soft targets, since peripheral, sorry, I said that wrong, nerves and spinal cord are extremely sensitive. Figure 3a shows a compact, lightweight, and stretchable device applied to the epidural space and the sciatic nerve in the mouse. Figure 3b shows heat maps of animals expressing a certain gene called CHR2, which demonstrates effective wireless opto-stimulation in the epidural space and the sciatic nerve. And Figure 3c provides a schematic illustration of a miniaturized device that uses a slightly modified energy harvester that's main purpose to aid and help for minimal disruption from joint rotation. Overall, there is a significant emergence of varying devices, both battery-powered and wireless, that are being researched in efforts to better understand the functions of biological processes and play a role in treating various diseases by reducing pain, stimulating paralyzed muscles, and etc. Okay. In conclusion, the emerging wireless technologies summarizing this article can, through their diverse modes of operation, create a significant new opportunities for research. Specifically, recent development allow for ultra-miniaturized, high-performance optoelectronic/microfluidic devices, wireless power delivery systems, and soft mechanics designing platforms that can be fully implanted and operated continuously for long periods of time without adverse effects on the smallest animal models. They can also, there's active programs and advanced technologies that combine sensing, stimulation, and high data rate information transfer, have the potential to enable closed feedback loops that can be responsive in real time to biological processes. The prospects are significant not only for research but also for eventual clinical translation in therapeutic devices and bioelectronic medicine. Significance is that it gives insights already gained by implementation of these methodologies with conventional technologies are numerous. Further progress will be accelerated by the development of advanced technologies for delivering optical and pharmacological stimuli and for recording relevant responses. The disadvantages include efficiency that depend strongly on relative orientations of the antenna pairs, interference and standing waves that occur due to the reflections from environmental obstacles and absorption associated with metal components, water, or moisture. The latter effects can be detrimental to systems that are fully implanted into the body, and they limit practical operating ranges to 0.1 to 3 meters. Another disadvantage is that traditional wireless hardware has some utility in such cases, but major disadvantages follow from complex connections, undesirable mechanics, large sizes, and weights, resulting in requirements for externally mounted head stages that can be cumbersome for small animal models. Some limitations are that the means for power supply are among the most challenging aspects for implementing these types of systems in small animal models. Approaches based on electrochemical energy storage, such as batteries and supercapacitors, consume significant space. They are heavy and often involve toxic materials, and they offer limited operational lifetimes. Another limitation is that it's particularly for small animal models. Weight becomes a primary concern where even 1 gram can be problematic for mice. Yeah, next one. Some further implications of this research can be used to support future clinical translation into therapeutic devices and bioelectronic medicines, and it can also be used to combat diseases and reduce patient symptoms. Thank you.
Good job, guys. So actually, just if you don't mind leaving your screen up for a second. So this is whenever I did journal presentations like this, we would sort of just go through the slides and sort of just like point out what was done well and what could have done better and stuff like that. So there's just a few pointers that I wanted to just point out so the next group can understand those. But overall, you guys did a really good job, and the study was interesting, and I really liked your presentation. I'm glad you guys did it. So the first thing is a general note for everyone. I don't know if we made this clear at the beginning, but so this is a review article, and what we should focus on in the future is a actual study, just so that it's like a clear what was done, methods, results, and like that's just more what we were going for. But this was still really well done. So just in the future, that's probably what you'll be researching more in journal clubs and in labs. That's probably what you'll be focusing on. So for future groups, just try and find that rather than a review article. And Rebecca is going to go more into detail about that later, but just wanted to mention that. You can go to the next slide or so if you wanted to. So again, you guys presented well, and I enjoyed. Okay, one thing I thought that was good too was, so I was nervous at first because you just threw out the term optogenetics, and it was great that in the next slide you really went through what that meant, what it was, because that's important. Like, it's when you have medical jargon or like scientific stuff, it's very important to like explain what you mean, just so it's like you can explain on a basic level, almost like the people you're talking to have no scientific background, so that everything you say is clear. So that was really good that you guys explained that thoroughly. One thing though that is important is maybe next time to have less words on the slides, and then just make it a little bit more interesting, like highlight certain important words with colors or bolded, stuff like that. And then if you want to go forward to another one. So, okay, one more maybe, sorry. So like, okay, this is really good that you guys included the pictures. just one thing I would do to make it even better is to separate them and sort of make it bigger so that we can see it. Like, one thing I was always told is that we should be able to read the text from the back of the room. And I know we're all at our computers, but I'm even having trouble reading those little words. So, like, either blink that, like, put white over it so that you can't see it, or make them bigger and visible, because otherwise it's just distracting. But I'm happy that those pictures were included. And yeah, otherwise it was really good. I thought the presentation was interesting, and maybe actually, I mean, this comes with learning a topic, is you sort of rely less on a script, read it less, and sort of just speak as if it's like a conversation as you're teaching something. But you guys did a really good job. Does anyone have any questions about the actual study?
I was just going to add, Serena summarized it really well. Also, just another tip, like, especially if you're presenting to like a panel of judges or just in front of other people, and like if you have an audience, typically they're going to ask about like specific slides. So what I was taught was to have like a references slide at the end. And then after that, I actually had like hyperlinks, like to go back to specific slides. So you can use hyperlinks like within your own slide and sort of like create like images or something that you can just click and it'll take you to specific slides. And then also, I know Serena touched on this, and it's definitely going to be a lot easier once you learn about a specific topic a lot more. But just to make like your bullet points a lot more concise, and then you can sort of verbally expand upon them, and you can also have like a script, but just to make it more like readable and easier to follow. And then also like slides with images like these, they're really good. But like she said, like sort of make the images larger. And honestly, I don't even think you need text on slides like these. Like, if anything, you could sort of create like animated annotations that you put like on the picture, and then you talk about it like as you go, if that makes sense.
Yeah, I think that's good advice. And sorry, I know it seems like we're only picking out these things, but like the point of this is to learn the right way to do it. And you guys did a really good job. We just want to make sure it's like those little things are perfect for the next time you do it. Okay, good job. We can move on to group 4. So group 4, wait, sorry, how do you pronounce your name? Sadhana? Wait, you're muted.
Sorry. Yeah, I'm here. Sana is at class, but we recorded it, so.
Yeah, I just want to know if you had anything to say while I bring it up. Go ahead.
I didn't have
Otherwise,
anything to say. Our study was about the use of just probiotics intervention in helping reduce maternal like postpartum depression, and then reducing eczema in newborn infants. So ours is related to maternal health.
okay, so I'll play that now. Just let me know if you guys can hear.
Yeah, you can start like the first 10 seconds. Like we were having trouble with the like.
I'm sure. No, it's no worries.
A pregnancy study, a double-blinded randomized, and everyone, me and Sadhana are presenting on probiotics in a pregnancy study, a double-blinded randomized study.
Sorry. Okay.
So our introduction is that the study focuses on chronic non-communicable autoimmune and inflammatory conditions. And for example, allergy diseases are of the largest group. Using probiotics, the quality of immune system increases, and researchers and doctors believe that a healthy gut in a pregnant woman will lower the transmission or just like allergies and eczema in offspring, and also other maternal postpartum conditions.
Yeah, so the overall purpose of the study is to investigate mothers who are currently pregnant and see if supplementing probiotics such as HN001 in pregnancy while having mothers breastfeed after giving birth could significantly then reduce the rate of eczema in infants by at least 1 year, and also help to reduce maternal rates of gestational diabetes or bacterial vaginosis before birth, and maternal depression and anxiety postpartum that can often occur after giving birth. So the methods of the study, first looking at the participants. So the inclusion criteria for the study required 400 pregnant women that were expecting infants at high risk of allergic disease. And those enrolled in the study had to be enrolled at the 14 to 16 week gestation mark. And the study obviously was completely randomized in deciding what treatment participants would receive. So participants either received the probiotic or they would be randomized to receive the placebo until delivery and then continuing that until 6 months postpartum. And then looking at the study design, we see that it's a two-centered, randomized, double-blind, placebo-controlled trial. So as a double-blind trial, the researchers and the participants don't know what treatment they're going to be receiving in order to ensure that the study is wholly effective. The interventions that participants received was the probiotic intervention administered during pregnancy and during breastfeeding. So the methods, first we're going to look at the primary assessment and then compare it to the secondary assessment and secondary outcomes. So the primary outcome showed development and severity of eczema and atopic sensitization in the first year of life. And then the secondary outcomes focus on the diagnosis of maternal gestational diabetes and bacterial vaginosis at 35 to 37 weeks gestation. And other outcomes included maternal weight gain, postpartum depression and anxiety, infant birth weight, and the rate of cesarean sections. And then the samples collected for the study included the maternal and infant fecal samples, maternal blood samples, cord blood and infant cord tissue samples, breast milk, infant skin swabs, and infant buccal swabs. So here, it's a little hard to see, but this is essentially one of the tables that represented primary outcomes and the following related outcomes. And the second table of the study represented the secondary assessment, which we see here ranges from 14 to 16 weeks gestation to infant at 12 months.
Okay, so for the data analysis, they use an intention to treat analysis to assess the effects of the probiotic. And chi-square tests were used to compare the proportion in each study group with detectable levels of immunological markers in breast milk. And then with those levels, t-tests were used to compare the groups if the data was log normal distributed. And if it was not, then the Wilkson rank test was used. And the results for the study, there were no solid results in our clinical trial, but because the study is still going on, but they believe that there is a benefit for pregnant women to use probiotics. But again, there's not a lot of evidence. So there's an ongoing discussion about pregnant mothers using ingesting probiotics. They believe that there is a growing benefit for that, and it's not a routine clinical practice. Doctors don't really recommend it that often to their patients, but it's growing. And in 2015, the World Allergy Organization guideline said that pregnant women whose infants are at high risk for allergy should take probiotics. However, like I said, this recommendation is conditional and based on low quality evidence.
So yeah, just going off of the discussion, looking at the significance of this study, If the study does show to be like successful, then it can show researchers and medical practitioners and those in the medical field the range of maternal and infant outcomes that can be expected and examined as a result of a single intervention of a probiotics. And if a probiotic intervention is adopted and does seem to work out, then it can be placed into medical practice with ease. And understanding probiotic action in exactly how it prevents gestational diabetes, infant eczema, and infant atopic disease could be really essential and really beneficial to the mother and her child. And lastly, if the study is successful, then it can just show that using the HN001 probiotic supplement is safe and has no harm or other effects to the mother and her child. And lastly, looking at just some of the limitations. So now with the current food market, there's many food and drinks that do contain probiotics, even if they're at small amounts. So this kind of makes it difficult for researchers, as the probiotics contained in food and drinks can't really easily be incorporated into study data collection. So this creates significant challenges because it makes it difficult for researchers to kind of tell if the probiotics that are impacting or affecting the mother and the child is coming from the food they're eating or from the probiotic intervention itself. So that kind of makes it difficult to gather that evidence and enable confident recommendations to be made in the clinical setting. So this was our study. I think the study was done in 2015, but we weren't able to figure out where the results were. And on the paper that we had, it just said that the study was still ongoing and that results weren't published yet. But yeah, so this was the paper we chose to do, and we referenced just this paper. So thank you guys so much.
Okay. That was a really good job. I liked the topic. I thought it was interesting. Another thing just about generally choosing articles, I would just try and choose one where there were results, obviously, just so you could talk about that. But it's no worries that that wasn't the case in this time. It's good that we saw that just so we could see in that case what you should do, because you guys did do a good job of sort of talking about how there were no results at the moment, but what you thought was going on. And you guys, actually both groups, did a really good job talking about the limitations of the studies. And that's always really important, because as a researcher, you should always look with like a critical eye. So that was a good job in both groups. I think one thing you also did that was good is you sort of did a great job explaining what type of study it was, and you went into detail about that. You didn't really leave any technical things up to question. You were great at explaining everything on a pretty basic level. One thing that both groups did that I mentioned before is just less words on the slides are better, and sort of, if you can, make it a bit more like appealing to the eye, and so that people will pay attention to the presentation rather than just zone out and listen to what you're saying. But you did do a good job explaining. And then with the pictures, I don't know where it was, but definitely try and, the chart. Yeah, I was just trying to say, like, same thing, no worries, but just make that a bit bigger. Like, something that you can do is always just reformat it yourself so that it's bigger font, or it just shows what you need it to show. But basically, yeah. Does anyone have anything to add? Any questions for the presenter? Any constructive criticism? Anything you liked? No. Okay. That's fine. So now that we've run through two sort of presentations, next time I think it's important to have some like feedback from the audience. So we'll go over that more next time, but we'll have everyone take notes and sort of just be prepared with at least one question or piece of criticism or something that you liked, so that we can all be active listeners and talk to each other and learn about each other's interests. I think we're going to skip the Kahoot for now just because it's pretty long and it's like just basic research terms. And I think you guys know it. And it's like, it's fine. We're going to save it just in case we want something else fun to do. So, next meeting, as I mentioned, we have groups 1 and 3 presenting, which is exciting. And then next week on Friday, November 13th at 2 p.m., we have our first event, which all of you qualify to go to, which is just really exciting. It's Dr. Chet Sherwood, who is a professor at GW, and his lab specializes in evolutionary biology. And specifically, his research is on the organization of human brains compared to primates. So, like, which sizes which brain regions are bigger where and what that could mean. So their lab is really cool because they sort of look at like the cross sections of the brain and analyze what that means and how it could have happened. And although they're not really in person right now, they're still doing research. So he's excited to talk to everyone. And again, we can practice this, but active listening, questions, and all that. So that'll be exciting. But we'll post more information as that, gets closer. As usual, office hours, me and Rebecca are willing to meet. If the presenting groups or anyone else needs help within the next week, it's going to be on our off week, but same time on Mondays at 6 p.m. Or if you have any questions, don't hesitate to reach out over email. We'll always be available to help. And then, Rebecca, is there something you wanted to say?
Yeah, well, we covered it a lot before, but I was just going to say, so a lot of review articles are actually really good sources, like for information, and you can use like extra articles like to add to your presentations. But the sole like purpose of presenting these articles is to present like a specific study that follows like the classic study design. So usually it's like intro, methods, results, conclusion, discussion. So I put a picture in on the left. It's just like if you go on a site like PubMed, I know other sources have this too. There's usually like you can sort of filter your search. So I selected free full text because sometimes you have to have like a subscription to actually access like the whole article. And then for the article type, we went over this like in previous meetings, but as long as it follows some sort of like experimental design, and if you want to have like review articles for additional information, you can like specifically click that. And then for publication date, typically within like the past 10 years would be considered like relatively recent. So yeah, and also just make sure that you let us know at least 3 days before so you have enough time in case there's any issue with the article that you chose.
Yeah, thank you. Yeah, and so that's really all for now. The groups that went today did a really good job. I'm excited that we had our first presentations, and I'm looking forward to our next ones. Our next meeting is not this Monday, but the next, and we'll have the research conference in between that. So, yeah, we look forward to that. And that's all for now, unless anyone else has anything to say or any questions. No problem.
Good job, guys. You all did really well today.
Yeah.